Welcome, everybody, to today's acid-base seminar. What I'm going to focus on today is the concept of whole body proton balance, and then discuss both type 1 and type 2 RTA. To understand these diseases, you have to have a conceptual understanding of what it means when the body maintains proton balance. So we'll start with that. So just again to refresh your memories, this is the Henderson equation. It's the non-log version of the Henderson-Hasselbalch equation. And this equation is important because it not only summarizes the acid-base chemistry of a bicarbonate buffer, but also the terms represent organ function. So the PCO2 in the numerator is determined by what your lungs do, and the bicarbonate in the denominator represents an analyte that the kidneys maintain at a concentration of approximately 24 milliequivalents per liter. So the question is, how do the kidneys do that? And the kidney has to perform two essential functions to keep the bicarbonate at around 24 millimoles per liter. The first is it has to reabsorb the bicarbonate that's freely filtered through all of our glomeruli. If the bicarbonate got filtered and nothing else happened after the filtration step, it would all end up in the urine. So that's the first thing that has to happen. The second thing that has to happen is that, and we'll talk about this in greater detail, every day your liver produces protons. It's like an IV. The hepatic vein is like an IV that puts acid into your body, and it's in the form of hydrochloric and sulfuric acid. And this can't be allowed to occur without something else compensating for it. And it turns out that the kidney actually makes bicarbonate above what it reclaims from the glomerular filtrate to match this daily hepatic proton load. And both of these things have to occur in order for the blood bicarbonate concentration to remain at 24. And we'll go into greater detail about both of them. So the first concept is the free filtration of bicarbonate. The plasma bicarbonate's around 24 millimoles per liter, and assuming we have a GFR of about 140 liters per day, you can multiply the 140 times the 24 to calculate how much bicarbonate comes through all our glomeruli, which works out to about 3.4 moles per day are filtered. As an example, there's about 5.4 moles of bicarbonate in a box of Arm & Hammer baking soda. So it's about 2/3 or so of the total amount gets filtered through our glomeruli. Now, if that was freely allowed to filter and nothing else happened, in about 4 hours, your blood bicarbonate would be zero. So clearly mammals have evolved to prevent that from happening. And what has evolved is a set of complex, what we call transport machinery in the proximal tubule. So this is a schematic of what happens. The glomerulus here is on your left, and the proximal tubule is shown in yellow, and on the right side is the blood compartment. So three compartments: the filtrate, the intracellular compartment, and the basolateral blood compartment. So the first thing that you have to know is that there's a protein on the apical membrane of all mammalian proximal tubule cells called NHE3. It stands for sodium hydrogen exchanger 3. There's a whole gene family of sodium hydrogen exchangers, and the one in all mammals is NHE3, and it absorbs one sodium ion and secretes a proton. Now, there's also a vacuolar proton pump that you might be more familiar with in the collecting duct that is also present in the proximal tubule. Most of the proton secretion, although occurs via NHE3, 80–85% of it. A smaller fraction of it is secreted via the vacuolar proton pump. And what happens is, when bicarbonate comes through the glomerulus, the acidification of the luminal solution from the protons that are secreted, the proton combines with bicarbonate to ultimately make water and carbon dioxide. The last step here of carbonic acid going to water and carbon dioxide is catalyzed by a protein called carbonic anhydrase 4. It's, again, a multi-gene family. There are many carbonic anhydrases. This one is attached to the apical membrane of the cell. It actually floats in the solution here, but it's attached to the plasma membrane. It's called a GPI anchor, and it catalyzes this last step. It accelerates it. It would occur anyway without the carbonic anhydrase. It just makes this last step occur faster. And then what happens is carbon dioxide that's created actually crosses the cell membrane. Some people think through aquaporin 1, the water channel, to get into the cell. So the carbon on bicarbonate actually doesn't get in directly. It gets in indirectly as a gas in the form of carbon dioxide. And then what happens is the reverse reaction occurs. Again, the initial step is catalyzed via a different carbonic anhydrase. This one is free floating around the cytoplasm, carbonic anhydrase 2. And ultimately, one gets bicarbonate, not shown here, and then carbonate that's generated in the cell. And it turns out it's the carbonate. The carbon leaves the cell, not in the form of bicarbonate, not in the form of carbon dioxide, but in the form of carbonate on a protein called NBCE1A that transports one sodium ion and a carbonate ion from the cell to the basolateral side, to the blood side. Now, this is called an electrogenic transporter. I notice some of the fellows call everything a channel. You should not call all transporters channels. Channels are very specific types of transporters that transport one ion, and they have very specific characteristics. So this is actually called a symporter or a co-transporter, and it transports one sodium and a carbonate ion. But because this carries 2 minus charges and this is 1 plus, it's called electrogenic because it actually transfers 1 negative charge from the cell to the outside. And because it does that, it actually can change its transport based on what the membrane potential is here. And remember that cells have a negative potential inside compared to the outside. And so it's the electrical potential here that is actually the driving force for getting the sodium carbonate out of the cell. It turns out that the sodium gradient and the carbonate gradient are actually in the wrong direction. The sodium outside is 140 here, and the carbonate is higher outside in the blood compared to the inside also. The sodium in the cell is around 20 millimolar. So if it was just the chemical gradients, you'd never be able to absorb carbonate in the proximal tubule. It's only the electrical driving force that exceeds the outward electrical driving force to drive the transport exceeds the chemical driving force inward, and that's why we get net transport out. The things that affect the membrane potential, though, will affect the sodium carbonate symport out of the cell. So that's how we reclaim bicarbonate from the lumen to the basolateral side. The second thing the kidney must do is match the daily hepatic proton load. So as I said near the introduction, the liver makes protons every day. On an average North American diet, it's around 50 milliequivalents of protons per day that are put into the body. And what happens is the kidney generates roughly the same amount of bicarbonate every day to match the daily hepatic proton load. And so the net reaction is protons come in from the hepatic vein, additional bicarbonate is generated in the kidney above what it reclaims from the glomerular filtrate. They're roughly equal, and that generates carbon dioxide, which you just get rid of via the lungs. So the 50 is just an average. It's not fixed, and it varies with what type of food we eat. If we eat a lot of eggs and hard processed cheese, we actually produce a lot of acid, a lot of protons. And if we eat a lot of spinach and raisins and most fruits and vegetables, we tend to generate bicarbonate from our diet. So depending on what we eat, that determines the net proton, or if you're a vegetarian eating raisins and spinach and vegetables all day, you're going to actually generate bicarbonate. You won't generate protons at all, and all the kidney has to do is excrete the bicarbonate load. And this changed also, our diets changed around 10,000 years ago when we went from being carnivores to herbivores, where we had more plants and vegetables in our diets, changed the net proton load. It turns out that if we have a ratio of animal to plant food in our diet of about 60%, the net acid production is 0. So when you go above that, you start generating net protons. And when you go below that, you tend to generate bicarbonate. So most diets in North America have a much higher animal to plant ratio than 60, and that's why we tend to generate around 50 milliequivalents of protons a day. And these protons are added to the systemic circulation. First, hepatic vein to the IBC. The proton will combine with bicarbonate to make carbonic acid, carbon dioxide, and water. The carbon dioxide will excrete. If we don't have a mechanism for compensating for this, then in about 2 weeks, our blood bicarbonate will be 0. You just have to calculate how much bicarbonate is in our body, in our cells, and in the extracellular fluid, and then every day you lose 50 milliequivalents. Well, it'll take about 14 days to deplete your entire body of bicarbonate if nothing else is done. So clearly that doesn't happen in normals. We maintain a blood bicarbonate of around 24, so this can't be the only thing that's occurring. And it turns out there's this process called new bicarbonate generation. And this is what happens. In the proximal tubule cell, glutamine, the amino acid, is taken up on the apical side and the basolateral side. I won't get into how that is occurring. There's specific transporters for this. And then in the mitochondria, it's converted into alpha-ketoglutarate, this organic compound. But in that reaction, 2 bicarbonates are generated, and also 2 ammonium ions are generated. The 2 bicarbonates leave the kidney via MbCE1A as sodium carbonate, as I mentioned before. And why do we care about the ammonium that's also made in this reaction? This is actually the source of most of the ammonia that ends up in your urine. It's mostly coming from glutamine metabolism. But you have to realize that when you're seeing that, in that process was also the generation of 2 bicarbonate ions that were not present in the glomerular filtrate. That's new bicarbonate which is being generated. So this bicarbonate then gets into the renal vein. You might think, okay, that's great, we'll compensate for our daily hepatic proton load. But the problem is the reaction also makes 2 ammonium ions. So the question is, if ammonium derived from glutamine were transferred to the renal vein with the bicarbonate, what would be the problem? The problem would be that the bicarbonate that you just sent into the renal vein that you would hope would match the daily hepatic proton load would actually be consumed in the urea cycle. Remember that the liver makes urea, and it makes it from 2 ammonium ions and 2 bicarbonate ions. That's how you generate urea. That's the net cycle in the liver. We won't go through all the steps, but basically you're taking 2 ammonium ions, combining them with 2 bicarbonate ions to make urea, carbon dioxide, and 3 water molecules. So if all the ammonia that was generated, the ammonium that was generated from glutamine went across the basolateral membrane into the renal vein, and it all went with bicarbonate to the IBC in the liver, you'd basically be turning glutamine into urea, 2 neutral compounds, and you wouldn't be doing anything as far as creating a net bicarbonate load to match the daily hepatic proton load. So clearly that can't be allowed to occur. And what happens is that, not shown here, there should be 2 arrows here, I apologize. Not all the ammonia that is produced in the proximal tubule gets sent to the renal vein or the urine. It turns out it's partitioned. There's a concept called ammonium partitioning, where 40% of the ammonia that's made goes back to the renal vein, and that depletes 40% of the bicarbonate in the formation of urea. So that's sort of a futile cycle. But we do get rid of 60% of the ammonia that was made, and we send it to the urine. It gets into the collecting duct first via these 2 RH proteins. This is the red cell RH disease in infants. These RH proteins are ammonia transporting proteins. They take ammonia from the interstitium. They're in the collecting duct and allow NH3 to get into the lumen of the collecting duct that then gets into the urine. We need to acidify the collecting duct lumen in order to create a gradient for NH3 to enter to be converted to NH4+. So we don't have all the ammonia that's made go into the urine. You might think that would be the best system, but we have 60% of it. So we lose about 40% of that bicarbonate that we made in the formation of urea. So it's not a perfect system, but we get rid of the majority. The other way we generate new bicarbonate is both in the proximal tubule and the collecting duct, and that's via protonation of phosphate. So protons are secreted via apical membrane proton pump, or in the proximal tubule via NHE3 and the proton pump. And any phosphate that's protonated in the lumen, we don't only protonate bicarbonate in the proximal tubule, we also proximate any buffer that has the appropriate pK. So phosphate is also protonated, and it's protonated in the collecting duct, but that proton actually, when it's secreted, causes H2CO3 in the cell to break apart and bicarbonate's formed. And if the bicarbonate is sent across the other membrane and not the same membrane as the proton, you'll actually, every time a proton is secreted, create one bicarbonate ion that is then sent to the blood. So this is the second mechanism for new bicarbonate generation that matches your daily hepatic proton load. So the mechanisms, again, for generating new bicarbonate are, first, you have to generate it from glutamine. It comes actually from alpha-ketoglutarate. This is a mitochondrial pathway. You generate 2 bicarbonate ions and 2 ammonium ions. A portion of the ammonia you send to the urine. Mammals, we don't send all of it like you might think teleologically. It should occur, but it doesn't. We do waste some of this bicarbonate by having some of the ammonia go back to the IBC and creating urea, but we prevent 60% of it from turning into urea. And then we convert HPO4 to H2PO4. Every time that occurs, you generate a bicarbonate ion that, when it leaves the basolateral membrane of the cells, acts as a new bicarbonate ion that was not part of your glomerular filtrate to match the daily hepatic proton load. This is also called titratable acid. You might see that in the literature. Okay, so that's what occurs normally, and some of these processes now are perturbed in RTA. And today I'm going to talk about 2 types of RTA. These RTA types were actually created by Tony Sebastian at UCSF. I think when he was a renal fellow with Curtis Morris, they started thinking about this typing. Unfortunately, it's a bit confusing because type 2 is the proximal RTA and type 1 is the distal. Type 3 is a combination of both, and type 4 RTA is really hyperkalemic distal RTA. Remember, it is not synonymous with hyporene and hypoaldo. That's only one of the many, many, many types of hyperkalemic distal RTA. So we'll talk about both the genetic and acquired causes of proximal and distal type 1. These are the hypokalemic RTAs we'll talk about. So just as far as the history of this thing, as far as when it first probably appeared in the literature, there was a paper in 1935 by Lightwood they talked about calcium infarction of the kidneys in infants, where it looked like there was calcium in the kidneys. And in retrospect, these are probably kids with distal RTA. The term wasn't used in the literature at that point. The first time the word renal tubular acidosis appeared in the literature was in this paper in the American Journal of Medicine in 1951, a paper by Pines. And they discussed what they termed renal tubular acidosis with osteomalacia. But now we know these diseases are a normal anion gap metabolic acidosis. It's an abnormality in either renal bicarbonate absorption or bicarbonate generation. And it's also important to know is associated with other electrolyte abnormalities. It isn't just an acid-base abnormality. There are changes in renal ammonium handling, calcium excretion. Patients are hypokalemic and they have abnormalities in water homeostasis, and they can have extrarenal manifestations that also, if you know them, give you etiologic clues as to the actual cause of the acid-base abnormality. So here's a 57-year-old female who was blind with bilateral glaucoma, bilateral cataracts, short stature, on exam, bilateral corneal opacities, abnormal teeth, and intellectual disability. So when you know the phenotype, this really can only be one thing. When you see it, you would know on an exam, if you know the phenotype, what this must be. But this shows the band keratopathy on exam, the abnormal teeth, especially the enamel, and calcifications of the basal ganglia. And these are the blood chemistries. The patient had a pH of 7.27, calculated bicarbonate of 12, and a PCO2 of 27. This is a non-GAP compensated metabolic acidosis. And the urine pH was 4.9. So the urine was acidic. and the fractional excretion was 30%. I'm going to talk about that because I think there's a misconception that patients, this is a patient with proximal RTA, the patients with proximal RTA have a high urine pH and bicarbonate in the urine, and that is not entirely true. We'll talk about the phases that one sees clinically in proximal RTA. Now, the fractional excretion also should only be measured when the blood bicarbonate is normal. The fractional excretion in these patients is very dependent on what the bicarbonate concentration is. And this fractional excretion was obtained when the patient was treated and the blood bicarbonate was returned to normal, which is appropriate. We'll talk about all those subtleties, but the fractional excretion can be any number, and the urine pH can be either high or low in patients with proximal RTA, depending on what phase of the disease you happen to see the patient with. So that's going to be important on an exam, and we'll get into that because that seems to be a misconception among some of the fellows. Again, the fractional excretion is calculated like we calculate the fractional excretion of anything. We take the urine concentration of what we're interested in, divided by the plasma concentration of what we're interested in, times the inverse, the plasma creatinine or the urine creatinine. And what this represents is the amount excreted over the amount filtered times 100%. So it really tells you how much of what's coming through all your glomeruli ends up in the urine. And for bicarbonate, it should be like 0.2 or 0.3%. There's almost all the bicarbonate that's going through the glomerular filters is reabsorbed. So the amount excreted compared to what's coming through in a normal person is extremely low. You never see a number like this. So I'll talk about now isolated proximal RTA. And if you see that, and it's genetic, it's autosomal recessive, and it's now known to be due to mutations in NBCE1A, the sodium carbonate symporter. There have been autosomal dominant cases by family described, but no one, or thought to be autosomal dominant, but no one still has worked that up to know what the transporter is. And it's not clear that that's a separate entity. It's just really unknown. So the only genetic cause of isolated proximal RTA that's been described in humans are mutations in NBCE1A. And the problem is the carbonate or bicarbonate cannot get out of the cell. So again, we have this transporter not working properly. It can't transport sodium carbonate out of the cell in these patients. We now know more about this protein. My lab studies this protein for a number of years now. It's a membrane transporter. It has 14 transmembrane segments. These are parts of the protein that cross the plasma membrane, and they're also parts of the protein intracellularly. This is called the N-terminus, and then there's a C-terminus. And there are mutations known throughout the transporter that have been described over the years that are either missense. Missense is when you have a single amino acid change that changes the function. Nonsense is when you have a stop codon. For instance, there's a Q29X mutation, and you just don't get any protein made after that, or frame shifts. This is what the transporter looks like. We described its original cryo-EM atomic structure a number of years ago. It's a dimer. You can't really tell much from this, but there are two subunits that are identical that interact, and each of them transports sodium carbonate. So it's a bit confusing because per dimer, you actually get a sodium let's say this is monomer 1, sodium carbonate gets transported via this one and this one. And we know a lot more about how it does the sodium carbonate transport. This is a paper we just published showing the mechanism of the transport. And it's actually quite interesting. This is one of the monomers, and the purple represents what's called the gate part of the protein, and the green is called the core. And it turns out that this particular protein actually works like an elevator. The green actually binds the ions, the core domain, and actually moves the ions from one side of the membrane to the other. That's why it's called elevator type shift. And it moves about 5 angstroms. So this is just a schematic showing sodium carbonate. It doesn't show the ions separately. It's very simplistic cartoon. But it shows the ion binding site here shown as an open sphere. This is OF stands for outward conformation. and the ions bind, and that causes the green actually to move. You can actually see the atomic structure, the thing moving after the ions bind. So it really is like an elevator. It binds and then it releases the sodium carbonate on the other side of the membrane. So elevator type movement of ions. There are many types of mechanisms how transporters, whether they're exchangers or whether they're symporters, move ions. This has nothing to do with what a channel does. A channel, nothing moves anatomically or structurally. You just have an open pathway and the ion moves through based on gradients. There may be amino acids that attract the ion, but you don't get movement or changes in the structure of the protein that like you do with co-transporters or symporters and exchangers. This is one of the interesting mutations. It's a T485S mutation. It shows the electrogenicity of the protein. As I said before, because sodium and carbonate carry one negative charge on patch clamp studies, these are some studies we did years ago, you can, when you put the protein in a bicarbonate or carbonate containing solution, you can see the current here that is transported across the membrane. This white here is in a non-bicarbonate solution. There's no current. But in the mutated protein, you never get the carbonate current. It always looks like it's in a non-bicarbonate solution. So the thought is that it actually doesn't transport carbonate anymore, but the carbonate transport is turned into bicarbonate transport because of the mutation. If that occurs, the protein will not be electrogenic because bicarbonate only has one negative charge and it'll be electroneutral. If it's electroneutral, it won't sense the cell membrane. And now the only thing driving it are the chemical gradients. And remember what I said before, that the blood sodium and blood carbonate or bicarbonate concentration are always greater than the cell. So if it loses the electrical driving force, which is how we get carbonate out of our cells in the proximal tubule, you'll only see this passive chemical gradients, and the cell just fills up with sodium bicarbonate. The patients cannot absorb carbonate at all because the mutated protein doesn't sense the electrical potential. So why do these patients get extrarenal manifestations? And that's because NBCE1 is not just in the proximal tubule, it's in many organs in our body. It's in the brain in a number of locations. So exactly what is occurring is not clear, but they can get migraines and intellectual disability. It's in many parts of our eye, and I'll show a little bit of that. It's required to keep your cornea, prevent the cornea from becoming opaque and the lens from becoming opaque. And it also affects your intraocular pressure. And it's essential for forming enamel in our teeth. Without NBCE1, you cannot form teeth properly. The ameloblasts need teeth in order to both shape the teeth and calcify the teeth appropriately. I'll just show a little example of the importance in the eye. Some of you may be aware that every time we have a pH on the front of our eyes in the tear film, and every time we close our eyes, the solution acidifies, because when our eyes are open, we're constantly losing carbon dioxide. And we have actually a respiratory alkalosis in the tear film when our eyes are open. But when we close our eyes, carbon dioxide can't leave and the pH drops, and it's very dynamic. It's very acute and dynamic. We're constantly changing the pH in the front of our eye every time we open and close our eyes. Now, when we have this occurring, when carbon dioxide is going from 40 inside the eye to 0 in the front, we have to prevent the front of the eye from alkalinizing. And that is done because we have NBCE1 on the endothelial cells on the inside of our eye that actually transports sodium carbonate out of our cells. And that acidic keeps the stroma here acidic. We need to do that because if we don't prevent this alkalinization, every time we open our eyes, calcium phosphate is going to precipitate. So remember, calcium phosphate solubility is very pH sensitive, not just in our urine when we think of calcium stones, but every solution, including the front of our eye and also the stroma of the eye. So the sodium carbonate symport here removes carbonate here and actually lowers the pH again back to normal. But these patients don't have functioning NBCE1. because of the mutation, and the front of the eye gets very alkaline, and they precipitate hydroxyapatite, and that's why they get the band keratopathy. And it's actually treated with EGTA tear solutions to bind the calcium. So again, they have impaired NBC1A transport. That's why they can't transport sodium carbonate. NHE3 mutations have not been described in man, although there are knockout mice, and they do have a mild phenotype. Carbonic anhydrase 2 mutations have been described in humans, and they get a proximal and distal RTA because the carbonic anhydrase 2 is important not only in the proximal tubule, but also the collecting duct. And TASC2 mutations have been described. It's a potassium channel. They've been described in mice, and they get a proximal RTA because when this channel is mutated, the membrane potential gets depolarized, and that prevents NBC1A from transporting as much sodium carbonate out of the cell. Remember, it's very membrane potential sensitive. Now, we can have isolated proximal RTA, or we can have Fanconi syndrome, where we have proximal RTA with other transport problems in the proximal tubule. Now, remember, Fanconi syndrome doesn't mean you have to have proximal RTA. You can have a number of transport problems, whether it's uric acid, phosphate, amino acid transport defects. If you see a bunch of those without bicarbonate or proximal RTA being present, you still have Fanconi syndrome. It's not specific for proximal RTA plus some other things. Any combination of proximal tubule defects that are more than just one defect, you can use the term Fanconi syndrome. And I won't get into all these other diseases and what the pathophysiology is because of time constraints. What I'd like to do is focus a little bit about the proximal RTA phases, which not everyone is aware of. Simplistically, people think with every proximal RTA, there's going to be bicarbonate in the urine, and that, as I mentioned before, is not true. So we divide it up into acute, chronic, and treated because we find different phenomenology in all these phases. In the acute phase, you haven't reached a new steady state yet. You just started dumping bicarbonate. Let's say you just gave someone acetazolamide or Diamox. The bicarbonate will fall a little bit, but it still exceeds the proximal tubules and the nephron's ability to absorb it. So we will have bicarbonate in the urine for the first time, and the urine pH will be high. And if we measure the fractional excretion of bicarbonate, at that point, even if we don't normalize the bicarbonate to 24, it'll come back high. But if we keep waiting, what happens is the bicarbonate gets dumped in the urine, and now you have a bicarbonate of 15, let's say, and now you're able to absorb that glomerular filtrate. It's a much lower load than before. It's not 3.4 moles of bicarbonate per day anymore. It's much lower, but your defective proximal tubule can now handle it. It's not that we lose entirely the ability to absorb bicarbonate. We lose a percentage of it. And so at that point, what happens is your urine pH will actually be low, and you're going to get a low fractional excretion of bicarbonate if you do it when the bicarbonate is, if you do your urine and blood measurements of bicarbonate and creatinine with a bicarbonate of 15. Now, if you come along and treat the patient and bring the bicarbonate back to 24, again, the urine pH will be high, and again, you'll see the high fractional excretion. It'll be a little bit higher than before because you have a higher blood bicarbonate here, so your filtered load is greater. So remember that these chemistries in the urine, as far as urine pH and the fractional excretion, are dependent on at what point you saw the patient. And we rarely see the patient in the acute phase. Of course, if it's genetic, we never see the patient when it was just created. We only get to see patients when it was just created if it was pharmacologically related, like a phosphamide or some drug that will cause proximal RTA, or Diamox, a carbonic anhydrase inhibitor. But we see the patients most of the time clinically when they've reached a new steady state, unless they've been treated. And once they've reached the new steady state, the urine pH is low. Your kidney's trying to acidify the urine like crazy. They have a severe metabolic acidosis, and their fractional excretion is now, back to what it is typically in a normal human. And then when you come and treat again, then you bring out the defect again. So remember that for the rest of your careers, that proximal RTA does not mean bicarbonate in the urine. That's only when it's treated appropriately or when you first just saw the patient. So what is the mechanism of the metabolic acidosis? It's a defect in proximal tubule bicarbonate absorption, and we diagnose it by measuring the fractional excretion, but not when the bicarbonate is now in a new steady state and low. We have to return the bicarbonate back to what it should be, around 23, 24, and then we do the fractional excretion. So always do it on a treated patient. Remove the cause, like ifosfamide or topiramate, the seizure drug. It's also a carbonic anhydrase inhibitor. And then we treat the patients. Children, they require more alkali because they're growing than adults, with sodium citrate or K citrate. We tend not to give bicarbonate because it creates CO2 in the stomach. And citrate is an organic anion that's converted to bicarbonate, just like lactate is in our PD solutions, just like acetate is in our TPN. Anything with carboxyl groups, when they're metabolized in the body, are converted into bicarbonate, just like the ketone bodies, when we treat ketoacidosis with insulin, the ketone bodies, the beta hydroxybutyrate and acetoacetate, their carboxyl groups are converted into bicarbonate. It's all the same concept. There are additional mechanisms probably in proximal RTA also, not just the loss of bicarbonate. There's probably also a problem with new bicarbonate generation from glutamine. And this is sort of a subtle idea, and there's indirect evidence for this. These are patients with the autosomal dominant group. Normal patients had a urine pH of 6, and then when they were given an acid load, the pH went to 5. The RTA patients were in a steady state, so they didn't have a high urine pH. They had a lowish urine pH, but then when they were given an acid load, it didn't really go up as much as the patients at baseline. The ammonia stayed low, and you can see in the baseline here too, even though they have a low urine pH, their ammonia excretion rate is not high. A normal person going from 6 to 5, look how much more ammonia there is in the urine. That's because the kidney generates more new bicarbonate from glutamine. You generate more new bicarbonate, and remember that reaction also makes ammonia, that 60% of it of which ends up in the urine. But in these patients, despite the urine pH being spontaneously low, they have the same amount of ammonia in the urine, and then when they're acidified, the ammonia goes up, but not what you might predict in a normal person. And also, there are mice where MBC1 has been knocked out, and what you see is, despite the fact that in the steady state they have a low urine pH, again, this is evidence that proximal RTA in the steady state, the urine pH is low, there's no bicarbonate in the urine, but look at the urine ammonia excretion, it's very low. Again, we're not measuring production, but it's evidence that the generation of ammonia is also low. Okay, let's switch gears now to a 3-year-old female with decreased hearing and polyuria, 18 percentile for the height, severe bilateral sensory neural hearing loss, and rickets. And these are the numbers: hypokalemia, a calculated bicarb on the blood gas is 11, non-GAP metabolic acidosis, compensated appropriately, and the pH is 7.1 in the urine. This is in the steady state with a high calcium to creatinine ratio of 1.2 and nephrocalcinosis in both kidneys. So again, if you see this, you're thinking of a group of diseases that involve distal RTA. So this is hypokalemic distal RTA. It involves the type A intercalated cell that I've talked about in previous talks. This is one of the cells in the connecting tubule and cortical collecting duct, and also in the outer medullary collecting duct that are involved in urine acidification. Remember, there's 2 types of intercalated cells. Another one, that the type B cell that secretes bicarbonate, but the problem is in the type A. So the transport machinery in the type A intercalated cell differs from the proximal tubule. Protons are secreted mainly via the vacuolar proton pump, which is a minority proton secretory pathway in the proximal tubule. There's also a minority pathway. This is called a proton potassium ATPase. It's the same transporter that transports protons into the lumen of the stomach and acidifies the stomach, but this is not as important in the type A intercalated cell. For every proton that's secreted, you generate a bicarbonate ion. The bicarbonate doesn't leave on NBCE1A, it leaves on another transporter. It's related family member, but it's not NBCE1A, and it's not sodium coupled. It's called AE1, anion exchanger 1. This is actually BAM3 protein. It's 60% of protein in the red cell membrane is AE1. This is a slightly different N-terminal variant, but it's the same transporter, and it exchanges one chloride for one bicarbonate. So these charges are equal, so it's electroneutral. It's not called a channel. It's called an exchanger. Transports a bicarbonate out of the cell and then returns a chloride into the cell. The chloride then recycles across this chloride channel called CLC-KB. You might remember this from the thick ascending limb. It's one of the transporters mutated in one of the types of Bartter syndrome. It plays an important role in thick ascending limb transport, but it's also present in the basolateral membrane of the type A intercalated cell. So these patients have a problem with the vacuolar proton pump. Now remember, the vacuolar proton pump is a very complex protein. It's made of actually 14 different proteins, 14 different genes that assemble into the final transporter. And it's divided up into two different domains. There's a V0 domain that does the transporting, and then there's a V1 domain that splits ATP. So it's anatomically broken up into these two areas. That you can picture the plasma membrane here. This would be intracellular. And in one of the groups of patients where mutations have been found is in the B1 subunit. And it prevents ATP from being split, and therefore you don't generate the proton to acidify the lumen of the collecting duct. And they have hypokalemic distal RTA. They have a high urine pH in the steady state. They have nephrocalcinosis, hypercalciuria, calcium phosphate stones, low citrate, and they have decreased ammonium in the urine. They also get rickets and osteodystrophy, and because the vacuolar proton pump is important for inner ear function and hearing, they have sensory neural hearing loss. There's also patients that have mutations in the A4 subunit, and these patients have similar phenomenology. However, their sensory hearing loss may be more mild, or they actually may not have sensory neural hearing loss. So if you have a patient that has all this and their hearing is normal, you know it's the A4 subunit, and that's because in the inner ear in some patients there's an A1 subunit that can compensate for the loss of the A4 function. There are also patients that have normal proton pump, but AE1 is mutated. So they can secrete the proton, but the bicarbonate that's generated cannot get out of the cell. And there's two types of these. There's recessive disorders and dominant. The recessive tend to be in the tropics. They're more prevalent, and the phenotype is more severe as far as all these findings. Whereas the dominant form is found more in Caucasians. It's much rarer, and the phenotype is less severe. In the mice, there are other models that have been found. There's a potassium chloride co-transporter that's been mutated, and these patients these mice get distal RTA. There's another mouse model, another protein called SLC26A7. It also has a distal RTA phenotype. And there's many, many other mouse models that have been generated where they have a distal RTA phenotype. I won't get into the different phenomenology, except to say that the FOXI1 transcription factor has not only been found in mice, but is now a cause clinically of distal RTA, familial distal RTA. This is a transcription factor in alpha in type A intercalated cells, and these patients have problems actually creating these proteins, AE1, beta-1 subunit. They have a number of different protein expression problems. The intercalated cell doesn't develop normally, and they have a phenotype of distal RTA. So it's just another cause you need to know in addition to the B1 subunit and AE1. There's now the FOXI1 patients. So back to whole body acid-base balance, because we're going to talk about another subtlety where we have on average 50 milliequivalents of protein protons put into the body and 50 milliequivalents of new bicarbonate generated mainly from glutamine and alpha-ketoglutarate metabolism to match that, to prevent the blood bicarbonate from falling. But there's a concept called incomplete distal RTA. And the thought here is that patients can handle or match the daily hepatic proton load, or they may be on a lower hepatic proton load because they're vegetarian and they have normal acid-base status. That's the key. They do not have a non-GAP metabolic acidosis, but they have other phenomenology telling you something is going on. So most of the time, they can handle the hepatic proton load, or the protons are being buffered elsewhere, and it's not manifest in the extracellular fluid as a drop in blood bicarbonate. So their bicarbonates are normal, their systemic pH is normal, but they have a low urine citrate excretion, nephrocalcinosis, recurrent calcium phosphate stones, and hypercalciuria. So they have calcium phenomenology and a low citrate. And then when you stress the system, it's like giving a water load. You might not see dysnatremia initially until you stress the system, or other stress tests that we do. In this case, we give ammonium chloride acid loading. Remember, when you give ammonium chloride, you acidify the blood, not because the NH4 dissociates. Its pK is much higher than 7.4. It's 9.2 or 3. 9 something. It stays as NH4 plus. It's just the ammonium that you're giving gets into the liver and consumes bicarbonate in the urea cycle. And so you acidify the hepatic vein more. And that's why ammonium chloride causes a non-GAP metabolic acidosis. But you can give this and you can dial in whatever bicarbonate you want in the blood. They get acidemic, and then a normal person, the urine pH should drop below 5.3 because you're acidifying the blood. And the kidney recognizes this and tries to acidify the urine. These people cannot. So that was sort of a historic way of diagnosing incomplete RTA, since Wrong brought this to the fore in 1959. Since then, now we have greater subdivisions of incomplete distal RTA. We divide the patients up into those that can't acidify below 5.3, but there is a subgroup that actually can, but they have additional phenomenology. So of the ones that cannot acidify, 48% also have decreased NH4 plus in the urine, but set in 17% of the total, they're normal. 35% of the patients can acidify and they have decreased ammonia. So it's a little hard to remember all this, but just know that incomplete distal RTA is not a homogeneous group as used to be thought of. It's been hypothesized that perhaps some of these people have mutations in the Rh proteins that let ammonia into the urine. If you knock these out in the mice, you see similar phenomenology where they can acidify their urine, but they just can't transfer ammonia into the urine normally. And then there's glycosphingolipid metabolism that depends on these two enzymes, UGCG and CST. And when you knock these out in mice, you also get a phenomena where there's less ammonia in the urine, but they can acidify. And it's thought that these glycosphingolipids in the interstitium of the kidney in normals bind ammonia and are important for getting ammonia into the final urine. And in these mice, they're lacking that. And so the kidney is unable to transfer as much ammonia into the urine. That's a hypothesis, and it's never been seen in humans yet. So the concept here is what happens in a normal kidney when you have an extrarenal metabolic acidosis, in order to understand what's not happening in distal RTA. So if I give you a proton load, let's say lactic acidosis or ketoacidosis, a number of things happen. First, you increase ammonia production from glutamine because your kidney's trying to make new bicarbonate to match the protons coming from the lactic acidosis. So you find that glutamine uptake in the kidney really goes up. There's more alpha-ketoglutarate metabolism, but that occurs with more bicarbonate generation, which is what you want. But the side effect is you make more ammonia. The kidney, though, takes that extra ammonia that's made, and now it sends 80% of it into the urine. So the kidney, in an extrarenal metabolic acidosis, changes its ammonia partitioning. Instead of 60% of the total going into the urine, it increases to 80, which is good because you don't want all that extra ammonia going back into the blood and making urea. Otherwise, this would be futile. Kidney also increases new renal bicarbonate generation from protonating phosphate in the proximal tubule lumen and the collecting duct lumen. As I said, we call that titratable acid, or TA, excretion. And the liver compensates by decreasing urea production. In a metabolic acidosis, there's less urea made. Your BUN actually can drop because ureagenesis decreases, which is also a good compensation because that will utilize less bicarbonate in the liver in the formation of urea, and it'll free up the bicarbonate that's coming from the kidney, the new bicarbonate, to match the extra protons that are coming in from the lactic acid or keto acid, or whatever the source of the acid. And these are the two problems in distal RTA. This change in urine to renal vein ammonia partitioning has a problem, and also the increase in new extra titratable acid generation and excretion don't occur. Yeah, we're missing the arrows again, unfortunately. But what happens here is that the ammonia that normally would be 80% now in the urine and only 20% in the renal vein, with distal RTA, you can't get that 80% in. You need to be able to acidify the urine in order to get ammonia from the interstitium to cross and go over into the collecting duct lumen. Remember, it's a passive process, and the proton secretion is important because when protons are secreted, you're lowering the NH3 concentration in the lumen of the collecting duct, keeping that driving force for ammonia to continually enter. And when you can't acidify the urine for whatever reason, whether it's a B1 subunit mutation or AE1 mutation, whatever the reason, you don't lower the urine NH3 concentration enough in the collecting duct outside the intercalated cell to create that driving force for NH3 to keep entering. So the ammonia that's generated in the kidney, instead of going into the urine, you have excess ammonia now coming from proximal tubule production going to the renal vein instead, and you have excess bicarbonate consumption in the liver and the formation of the urea. So the cause of the metabolic acidosis, one of the causes in distal RTA, is excessive urea production, inappropriate ammonia partitioning to the renal vein. If someone says to you, why does my patient getting distal RTA? It is not, as some people think, the lack of proton excretion in the urine, because the amount of protons you actually excrete free is micromolar per day. Remember, with a urine pH of 6, you have 1,000 micromoles or 0.1 millimoles of protons in your urine in every liter. So you're only getting rid of 0.1 millimoles a day when your urine pH is 6. We don't get rid of protons free in solution. So that's not why in distal RTA your blood gets acidic. It's because you have inappropriate removal of ammonia in the urine. The ammonia has to get out of the kidney. It has to get out either into the renal vein or urine if it's being produced. And if it can't get into the urine, it gets out the renal vein, and then that goes to the liver, and you drive the urea cycle excessively. The other reason is that you don't secrete protons normally, again, to protonate phosphate. So your titratable acid excretion is lower than normal in distal RTA. So two reasons why you have a problem. The first problem is you make the bicarbonate, unless there's a proximal tubule, as some people think, generation problem. It's a hepatic consumption that's a problem. Whereas here, with titratable acid, it's actually a generation problem of the bicarbonate. So you probably have heard of looking at the ammonia in the urine as sort of an indirect way of assessing whether someone might have distal RTA. And it's extremely indirect because remember, the ammonia in the urine depends on generation and also a whole bunch of transport steps to get to the urine. And also there are other reasons, clinical nephrology, why the urine ammonium is low. For instance, in CKD or patients, the urine ammonia is low, not because there's a partitioning problem between the collecting duct and the renal vein, but the ammonia isn't generated. Glutamine uptake in CKD is very poor, and that's why actually patients with CKD get a metabolic acidosis. They can't generate new bicarbonate normally from alpha ketoglutarate. But when that process is decreased, they also don't generate as much ammonia. So the low ammonia in the urine doesn't mean you have distal RTA. It's just one of the many causes. And we used to have these indirect ways of measuring it, either the urine anion gap or osmolar gap. Now at UCLA, after a number of years, we can finally convince the clinical lab to measure it directly for us, which we can do. What people don't realize also is that because of the partitioning issue, when ammonia doesn't get into the urine, especially when the distal RTA is just created, it actually ends up in the blood. And so if you have someone with elevated ammonia in the blood and low in the urine, that really can only occur with distal RTA. It's a clue that you have distal RTA, and most clinicians are totally unaware of the literature. It's when it was first created, because once you treat the patient, you affect the ammonia and the bicarbonate partitioning and production of both. But at time zero, if you happen to be there, you'll find a high blood bicarbonate and a low urine bicarbonate. It's the same thing that happens with Diamox. If you give someone Diamox, you raise the blood ammonia level and lower the urine ammonia level. We have a few minutes left, so I'll go quickly here. As far as the hypercalciuria, as I mentioned, they get nephrocalcinosis and calcium phosphate stones. It was known from dog studies that as you increase the blood bicarbonate, the urine calcium decreases. Most clinicians feel simplistically this is due to decreased bone dissolution, but that's not the mechanism. As I'll show, we don't have hypercalciuria because the blood pH is low and we're dissolving bone. The calcium is actually coming from a kidney transport defect. So we treat the patients with K-citrate, and we may or may not give thiazides to try to get the bicarbonate to around 22. We try to normalize the urine calcium excretion by elevating the blood bicarbonate. We also increase citrate excretion when we do this. But if the urine pH goes above 6.5, we hold off, and then we may give thiazides. That problem is with thiazides, you can make the hypokalemia worse. So the hypercalciuria in distal RTA and most patients with metabolic acidosis is felt to be the TRPV5 calcium channel. It actually is a channel in the apical membrane of the part of the nephron called the DCT2. There's distal convoluted tubule 1 and 2. This is the second portion, and the connecting tubule. It's in the apical membrane, and it takes calcium into the cell. And it's known that if you create a metabolic acidosis, you significantly decrease the expression of this transport or this channel, and you don't absorb calcium normally. Acidemia also affects the function of the channel, and when the channel sees a lower pH, it actually transports less too. So it's an expression and transport problem. So this is a normal mouse with normal TRPV5 looking at the urine calcium, and when you give ammonium chloride or acetazolamide, the urine calcium goes up. You might say, oh, that could be coming from bone. But when you look at a knockout mouse, first, the basal calcium excretion rate is very, very high, showing you that the channel is important for calcium absorption. But when the same mice are given ammonium chloride or acetazolamide and their blood is acidified, there's no change in calcium excretion. So this is good evidence that it's not coming from bone at all. They have a high baseline, and they're insensitive to metabolic acidosis as far as altering their calcium excretion. So the take-home message here is, don't in a knee-jerk way say that my patient is excreting more calcium because their bones are dissolving. Think of the TRPV5 calcium channel being affected. There are other subtleties here. Whenever calcium is in the urine, the kidney compensates by putting less aquaporin 2 into the apical membrane, and they actually get like a nephrogenic diabetes insipidus, but that helps keep the concentration of calcium lower than it would be. And it also, when calcium is in the urine, these are via the calcium sensing receptor, which is also on these cells. You stimulate proton secretion and acidify the urine, which is great because you don't want the calcium in a high pH environment. Patients with distal RTA are missing this. So the thought is that this is contributing to the calcium phosphate stones. They can't acidify their urine in response to excessive calcium in the lumen. They also have hypokalemia. I'll just show these causes, none of which have been proven. No one understands why patients with distal RTA are so severely hypokalemic. Now, as far as diagnosis, there are ways better than the indirect way of measuring the urine ammonia. If you're interested, you should give Lasix 40 milligrams with Florin-F a milligram. And in a typical patient or a normal person, the urine pH drops below 5.3 after 4 hours. I won't get into how Lasix does that today, but when you give Lasix acutely, you acidify the urine. And that's associated also with enhanced ammonia excretion in the urine. And so what's done is you give the Lasix and then you measure both of these after the patient has avoided initially, so that you're not collecting urine prior to that. Obviously, it's better if they're catheterized, but few are. And then there's another more historic test that was done, and that's to measure the urine PCO2. So the PCO2 in the urine actually is diagnostically important, and it's been known for at least 40 or 50 years that if you give bicarbonate and give a lot of bicarbonate and flood the collecting duct with bicarbonate, the PCO2 in the urine goes up about 30 higher than blood. So if I give any of you bicarbonate and get it high in the urine, your PCO2 in your urine will go to 70, and that is felt to be a reflection of proton secretion. In patients with distal RTA, it stays the same as the blood, or very close. It stays at 40. It's called the U-B PCO2 test. But nowadays, Lasix is really the definitive way to do it. And because of the time, I'll stop there. So does anyone have any questions about anything I said? Again, try to understand proton balance, the two main mechanisms as to how the proximal tubule and the collecting duct together keep the blood bicarbonate at 24. And then look at distal RTA and proximal RTA as experiments of nature where the normal physiologic processes clinically are perturbed, leading to the metabolic acidosis. And I'd be happy to answer any questions offline if any of you have any. So no one has any questions? Okay, everybody, that's it for today. Have a great rest of the day. Thanks.